Namaste, friends. Welcome you all for the biology session. Today, in this session, we're going to discuss about the chapter body fluids and circulation. We know that what exactly the circulatory system. Circulatory system is also called as circulatory system, which is also called as blood vascular system. Circulatory system, which is also called as blood vascular system. In this system, mainly what is that? Almost all the important functions like distribution of nutrients and transportation of oxygen to all parts of the body, collection of waste and harmful products, and that will be what is it? They sent to the excretory organs, transported to excretory organs, and where what is it? These waste product will be eliminated. And it also helps in the distribution of hormones and many other functions. Circulatory system, which includes the organs, mainly heart, I can say that which is a pumping organ, heart, blood, lymph, blood vessels. In some organisms, generally in the lower organisms, we can say that the circulatory system is also called as water-vascular system, or you can say that the gastrovascular system. It is based on the medium we can classify. So, circulatory system, which includes pumping organ, heart, blood, lymph, and then blood vessels. So, before going to take up this, what is it? The complete circulatory system, human circulatory system. We can know some of the the important things in the lower organisms, how this will be, and what type of the circulation are there. How we can classify the circulation into different types. Yes. So first thing here, we have to keep it in mind that body fluid. Body fluid. It is not like that only blood which acts as a body fluid, right? So along with that, we know that lymph, which is also acts as a body fluid, and we can also say that the lymph, which is also called as tissue fluid, and there will be urine, which acts as a body fluid, bile, sweat, right? Bile, sweat, and we can also say that the fluid which is present between the joints. You can say that the fluid which is present in the synovial cavity. Fluid which is present in the synovial cavity. So fluid in the synovial cavity, that is between the joint, right? Synovial cavity. We can also say that synovial fluid. Synovial fluid. Now, so body fluid which will going to circulate. How they are going to circulate? Circulation means, what is that? We can say that they continuously, what is that? In case of these body fluids will going to continuously circulate, or you can say that flow, continuously flows throughout the body. So that is why we'll say this, the circulation means flow of body fluid within the the body, right? We can consider as a circulatory, which constitute all those organs together, and the flow of these the body fluid, which includes under circulatory system. What exactly this the blood which includes? So blood mainly, it is called as a connective tissue or a fluid matrix. Connective tissue, we can also say that the fluid matrix. So this is the extracellular, right? It is a structure of connective tissue which is the extracellular fluid. And why we will consider this as the connective tissue? Here, the cells which are there, why we will consider this as a connective tissue? Here, the cells which are there, they are suspended. Cells are suspended in a liquid medium. ಇದು ಲಿಕ್ವಿಡ್ ಮೀಡಿಯಮ್ ಅಲ್ಲಿ ಇರೋದ್ರಿಂದ ದಿ ಸೆಲ್ಸ್ ವಿಲ್ ಸೇ ದಟ್ ಜನರಲಿ see whenever we're going to say that if you want to consider the fluid connective tissue, there will be a cells, right? Cells, group of cells will be there, hence we'll consider as a tissue. If you want to say that the liquid connective tissue, that is there is a fluid, that is a liquid medium. So suspended liquid medium, or we can say that the liquid matrix, which is called as plasma, that is blood plasma. In this blood plasma, these cells are present. So we may get question fluid matrix, that is may get the question fluid matrix of blood. Fluid matrix of blood is the plasma. Plasma is the fluid matrix of blood. Next, before going to know about this, the function of the blood, we can have, there are different types of circulatory system. So based on medium, first, that is based on medium. In some water will be acts as, in some water will be acts as the circulatory medium. So in that case, we can say in the lower organisms, where you can say that the water vascular system, we can call it as water vascular system. And in higher organisms, which we call it as blood will be there, blood vascular system. Blood vascular system. In this water vascular system, mainly which includes in sponges, we can say hydrovascular system. Sponges, we can say that hydrovascular, the system, or you can say water vascular system. And same like that in cylinderates, like hydra. In cylinderates, like in hydra, we can call it as gastrovascular system, because it will be flow to gastrodermis, that is gastrovascular system. And in case of the starfish, that is echinodermata, starfish, as this water which will going to move through ambulacral system, that in the each the radiated arms, we can see that in each radiated arms we can see the structures here, like this in the starfish, that is pentaradiated arm which will be there. So in this there will be a groove, right? Inside there will be a groove. These grooves are called as ambulacral groove or ambulacral system, hence it is called as ambulacral system. In the starfish, water vascular system is called as ambulacral system. So now in the blood vascular system, we can see the different types. Always it includes heart, but sometime there won't be a blood vessel. Larger blood vessels will be there, right? And blood vessels, sometime it will be divided into includes, or you can say divided into arteries, veins and capillaries. Arteries, veins and capillaries. So here heart, blood vessels includes arteries, veins and capillaries. If all these organs are included, if blood flows through these closed vessels, then we'll say that is a closed circulatory system. Closed circulatory system or vascular system. If it, there will be, heart will be included, but only larger blood vessels, larger blood vessels, no if no other the smaller vessels, then we will say that is open circulatory system. So in closed circulatory system, blood always flow, or we can say that it will always flow through the closed the tubes. We can't be able to see that the blood which will be flow within the body, that is always in the closed the vessels. Here the blood which will be pumped by the heart, and then that will be pumped through the larger blood vessels, and it will reach the body spaces where organs are present. So organs are bathed in the blood, and when this this space gets filled, so this space is called as sinuses. Body spaces are called as sinuses. So in these spaces, when blood gets filled, and it will move to the next place. So here blood does not move through, always does not move through a closed vessels. So that will be moved to the open spaces, that is the body spaces, hence it is called as open circulatory system. And here note, the closed circulatory system was discovered by William Harvey. We can expect this question. Next, let us move on to this. Here in the blood vascular system only, the closed and open circulatory system. Let me take the example for open circulatory system. So here in almost all invertebrates, open circulatory system, right? We can also say that open circulatory system where always blood flows through the blood vessels. So here the fishes, invertebrates, we can say, right? So invertebrates and fishes. And here in the mollusks, mainly it includes non-cephalopods. Not all mollusks are the open circulatory system, only non-cephalopods. And closed circulatory system. In the closed circulatory system, we can say that it includes reptiles. It includes reptiles, higher animals, that is reptiles, amphibians, and along with that it also includes birds and mammals. So in this case, in the closed circulatory system, we have two types. Here, single circulation. Single circulation means once the blood which will be pumped by the heart, right? If you consider this just for a diagrammatic representation to understand the concept, if once it is pumped by the heart, right, it will going to reach the the gills for oxygenation purpose, then it finally reach the body organs, directly to the body organs. So here only once it will going to, example in fishes we can see single circulation. So heart receives always deoxygenated blood. Heart receives always deoxygenated blood, hence that heart is called as venous heart. The heart which receives oxygenated blood, that is always blood which will be with carbon dioxide, more carbon dioxide, that is venous blood. And here in the double circulation, right, in the closed circulation, we have single circulation and one more which is double circulation. So in this case, double circulation, we have complete double circulation and incomplete double circulation. Complete and incomplete, I think we are all familiar with this. Complete means the birds and mammals, where it will going to the float twice before reaching the tissue, that is before reaching the tissue cells or organs. Here that is 4 chambers which will be completely, in this case the blood won't get mixed, always which receives the deoxygenated blood, and that will be pumped to the lungs, right, where oxygenation will be done, and that will be pumped back to the left auricle and then to the left ventricle, and here from to the all organs. So once it will going to receive, it will move to the lungs. From the lungs, once again it will come to the heart and then to the organs. So before going to reach the organs or tissue, it will going to the pass through the heart twice, hence it is called as double circulation. So from once it receives, that will going to pump to the lungs, and from the lungs after oxygenation it will going to reach, once again it will receive, and then from there it will pump to the organs. So twice it will receive. So here no the mixing of oxygenated and deoxygenated blood. Here no mixing of oxygenated or deoxygenated blood. Here as it has, what is it, the 3 chambers where it will going to get mixed. Here it receives deoxygenated, and here it receives oxygenated. Both will be mixed with the ventricles. We know that chambers which are there. In fishes, 2-chambered are, what is it, incomplete, we can say reptiles and amphibians. Incomplete, what is it, the in amphibians we can say 3-chambered are. In reptiles, incomplete 4-chambered are. Incomplete, except incomplete 4-chambered are, right? And in the crocodiles, which is a complete, hence it will going to show in case of, what is it, the double circulation. In case of double circulation, right? So example for the complete double circulation, right? Complete double circulation. So this is very, very important. That is single circulation, double circulation. Single circulation in fishes, double circulation in reptiles and amphibians. In reptiles and amphibians, blood gets mixed, hence it is called as incomplete double circulation. In birds and mammals, blood won't mix, it is called as complete double circulation. In the complete double circulation, exception is there, that is in the complete double circulation we can say that crocodile, right, even though it is a reptile, there we can see, what is it. And one more thing here, as I mentioned, that the non-cephalopods which are, which is going to show open circulatory system, cephalopods which is going to show closed circulatory system. Cephalopods, right, which show closed circulation. Closed circulation, even though these are the mollusks which show closed circulation. So next, move on to the blood content, how it will be. Here, based on this, we can also classify the heart, which is there. Once if you're going to receive, what is it, only deoxygenated blood, in short form, actually we won't represent this DO2, right, deoxygenated blood, then we'll say such heart which will be call it as venous heart. If it receives both oxygenated and deoxygenated blood, then we can say that such type of heart which is called as arteriovenous heart, right? Arteriovenous heart. So next we can take up the content of blood. What is the composition of blood? So here when we're going to know about the blood, the main thing you have to keep it in mind that blood components are mainly includes plasma, and so blood which includes, we know that the plasma which forms about, the 55% of the total blood, whereas the formed elements. In this we're going to get question, please focus on this. 55% plasma idare bloodalli, remaining 45% is because of the formed elements. So what is that plasma includes? Let me take this, what is it, water, majority of that which will be water, that is 90 to 92% water will be there, and proteins, there will be 6 to 8% proteins. These proteins which are very important, which includes fibrinogen, and then albumin, right, albumin, and along with that globulin, albumin and globulin. So these are very important, we're going to discuss about this later. Next, and then after that, almost all the ions, whatever we have discussed, that is the calcium ion, sodium ion, potassium ion, magnesium ion, chloride ion, right, and then carbonate ion, right, in the respiratory system we discussed, right. So all these along with organic compounds, which will be there in the blood. So here the one very important thing you have to keep it in mind, that the role of, this fibrinogen which helps in clotting, albumin helps to maintain osmotic concentration of the blood, osmotic concentration in the body fluid, or I can say of the blood, and globulin, which immunoglobulin, which provides, immunity, which play a role in providing immunity. So plasma without this clotting factor, if no fibrinogen, plasma minus clotting factor, that is what is that, fibrinogen, which is called as serum, so these are the things you have to keep it in mind. Next, I'll come to formed elements. Formed elements are generally two types, so I will write here. So in this case, formed elements, right, mainly it includes what is it, the agranulocytes and granulocytes. So let me write like this. First, granulocytes, which includes granulocytes because they have granules. Granulocytes and agranulocytes. Granulocytes means there will be granules will be present within the, what is it, the blood cell, within the blood cell. So here in detail we can discuss about this. So before that, let me give one simple chart where we can, what is it, know about the different types of cells. Granulocytes which includes three types of cells: neutrophils, basophils, and eosinophils. And these includes monocytes and lymphocytes. In this, the question we're going to get here, the nucleus are generally lobed nucleus will be present, that will be vary in number. Here no lobed nucleus. In both of these, what is that, no lobed nucleus, and here no granules are present here with, what is it, the granules, cytoplasm with granules are present. Now, so this is the very important thing. In case of, what is it, the, before going to, what is it, the know about these formed elements, in this we can say granulocytes, agranulocytes, which comes under, what is it, generally WBC, because these are the important, as I mentioned. And we have generally, in case of formed elements, we have three types. One is, what is it, the erythrocytes, other one is leukocytes and platelets. So here we can say that the red blood cells, right, RBCs, which are also called as red blood corpuscles, and then WBCs, white blood cells, which are also called as leukocytes. These are called as erythrocytes, and these are called as leukocytes, and here these are called as platelets. So platelets, which are also called as thrombocytes, play a major role in clotting. If their deficiency is there, no proper clotting will be takes place. Excess bleeding will be there. No proper clotting, that leads to excess bleeding, right? So excess bleeding. So here erythrocytes, just let us have, what is it, the important, these are the most abundant. I think you're all familiar with that. Most abundant, what is that, 5.5 million to, generally we can say 5.5 million to, what is that, 5 to 5.5, up to, and an average we can say 5 to 5.5 million, what is it, the cells will be there per cubic millimeter of blood. That is, we can say, right, so per cubic millimeter of blood, right, per cubic millimeter of blood, these many cells will be there. These are most abundant, and least abundant are leukocytes. So here the simple thing we have to keep it in mind, always they're going to ask, these are the least abundant. Leukocytes are the least abundant, means they will be in 1000, that is 6000 to 8000, or you can say that 6 to 10,000, within that they will be. And what is that here, this is a total, what is that, the RBC count. This is a total RBC count. So total number of RBC per microliter, is this one, that is total, what is that, blood count. So total red blood count we can say right. So here no nucleus, no nucleus will be there, and generally these are, what is that, biconcave cells, and lifespan is 120 days, and these will be destroyed in spleen. That is why the spleen is called as graveyard of RBC, where the destruction, damaged and infected cells will be, what is it, destroyed. And in case of, what is it, the WBCs, so we can see the many different, what are the types that is already mentioned. In case of WBC, we have neutrophils, basophils, eosinophils, and monocytes and lymphocytes. Let us have a look related to this. And here WBCs are less than a week lifespan. Lifespan is very less, and they're going to, what is it, the fight against the immunity. So now let us have some question. Before that, I will mention what exactly the WBC which will be there. So just let me take, this one. Here we may get the question related to, I will give one chart related to these things, right? So just you can listen to that, where here in case of, the leukocytes, right, means WBCs which will be, no hemoglobin, hence they are not, red in color. RBCs with hemoglobin, hence they are red in color. Hemoglobin, which imparts a characteristic color to the red blood cells because of the hemoglobin. That too, iron containing pigment, that is respiratory pigment, iron containing respiratory pigment. So WBCs. So here the very important thing, this count will be 6,000 to 8,000, cubic millimeter of blood, very less compared to these three. RBC, WBC, platelets. We'll take up the platelets later. So in this case, the asked question is that the abundant will be neutrophils. Among those, abundant will be neutrophils. They form 60 to 65% the total neutrophils, and then basophils. So here basophils generally, which will be, the 0.5%, 0.5 to, the 1%, and these basophils play a major role in the secretion of, make a note of this, histamine, and then we can say that the heparin, which will be produced, and then along with that it also, it produces a serotonin. Like mast cells, it will also produces the basophils, which produces histamine and heparin. So histamine is anti, generally we can say histamine, which is, not anti, this one, that is inflammatory, right, and which causes inflammation. And heparin is anticoagulant, right, so which acts as anticoagulant, which prevents the blood clot, right, anticoagulant. So these are the things you have to keep it in mind in case of neutrophils and basophils. And neutrophils, which will be there, generally 5 to 7 lobed nucleus will be there, and here in case of basophils we can see 3 lobed nucleus, and in case of, the eosinophils play a major role, in inflammation. You can say that they will play a major role in inflammation, but before that we can say, that is allergic reaction, which causes allergic reaction. During allergic reaction, their number increases. These are, generally which forms 2 to 3%, that will increase up to 60% within the body during, the infection, that is allergic reaction. So that is the very important, and it also helps generally, initially it helps to resist the infection. When it can't be able to resist the infection, other cells has to be helped, right? So hence it will going to create more, allergy. So these are the things you have to keep it in mind. And here this is 5 to 7 lobed, and this one is a 3 lobed, and eosinophils are 2 lobed nucleus. So this question we may expect. Next, here one more important thing, I will finish these things completely, and then these are the granulocytes what we have discussed, and agranulocytes, monocytes. Monocytes are the, generally these are the largest among all those, what is it, the leukocytes, monocytes, right? Monocytes are the, what is that, largest in size among those leukocytes. And these monocytes, in which generally, what is it, these, they play a major role in the phagocytic, and these, what is it, the monocyte which is going to produce further, what is it, the development, they are going to develop into macrophages, right? Monocytes develop into macrophages. And lymphocytes, there are two types, T and B lymphocytes provide immunity. So this is the enough for that, and if, what is it, the time permits, we can discuss further. So let us take open vascular system. Open vascular questions, that is, open vascular system is found in. So we discussed that generally whenever we're going to say that whenever the open vascular system, we can say that the invertebrates. So, and along with that, in some it will be, what is it, the, in case of earthworm, we can say that the closed, that is one which belongs to, what is it, annelida, right? And in invertebrates, not all invertebrates, exception is there, that is, what is it, the annelida, and also in case of non-mollusks, I can say that cephalopods. In case of mollusks, what is that, cephalopods and annelida, which shows, what is that, the closed circulatory system. Hence we won't select earthworm and man, we know that there's closed circulatory system. In fish also we can see closed circulatory system, whereas in prawn, right, that we're going to see open vascular system. Next, the advantage of closed circulatory system. So as we know that whenever the closed circulatory system, it maintains the efficiency, it can increase, right? So if need increases, these blood vessels constricts, and they're going to increase the blood pressure. Flow will be, what is it, the faster. They can, what is it, meet the need of the body. Hence we can say closed circulatory system, what is that, is very efficient, mainly to support the metabolic activity and to meet the need or requirement of the body, right? Requirement of the body. And one more thing here we can say, whenever these, what is it, the circulatory system, which will be closed circulatory system, that mainly helps, what is it, it generates a sufficient BP, right? That is what I mentioned, that flow will be faster because it generates sufficient blood pressure because of those vessels, right? It generates sufficient blood pressure and regulate the flow of blood. These blood vessels regulate the flow of blood so that easily they can meet the need, higher levels of metabolic activity, can support higher level of metabolic activity. Closed system can direct blood to specific tissue. No, it is not to the, what is it, yes, we can consider not to all tissue, that specific tissue. Yes, see, I will tell you, like kidney, if it has to receive more blood, it is going to, what is it, they receive more, right? Whenever we're going to say that, not all, that is not for all the tissue, to a specific tissue it can pump the blood, correct? So like heart, if blood pressure increases, heartbeat increases, same like that. So whenever the heart has to increase, what is it, the pressure, the volume will be going to increase. So blood volume increases, heartbeat increases. So whenever it has to decrease, it will going to play a role. Directly it can direct the blood to a particular tissue, correct? So exchange occurs more rapidly. Yes, so here we can see that whenever this blood flow which will going to takes place, that is the, what is it, these are the arteries, and that will be get exchanged with the capillaries, and that will going to regulate the flow, and flow will be very fast, and exchange occurs more rapidly, right? Where the carbon dioxide will be, what is it, the taken from the tissue, and to the tissue oxygen will be given. So that is one function of the circulatory system. It helps in the transportation of, what is that, respiratory gases. Next, closed circulatory system support higher level metabolic activity. So all these can be done by this closed circulatory system. So in open circulatory system, no, what is the advantage? Why? Because it won't generate any sufficient BP, or you can say that it won't maintain a rapid flow. There will be slow, blood flow will be very slow. Hence, closed circulatory system is, what is that, highly advantageous than open circulation. Which of the following set of animals has an incomplete double circulation? So we discussed that incomplete double circulation will be there in mainly, what is that, reptiles and what is that, amphibians. So incomplete, what is that, double circulation, because here both oxygenated and carbon dioxide blood, that is deoxygenated blood, will be gets mixed up because of those 3-chambered heart which will be there in the amphibians, and incomplete 4-chambered which will be there in the reptiles. So answer for this is, we can say, that is toad and lizard, right? So toad means which belongs to amphibians, and lizard that belongs to reptiles, whereas shark, right, which is not generally, we can say that the whale, that is a mammal, right? So here in this case we can see complete double circulation, right? So in case of whale we can see that generally, and the lizard, right, here in these fishes we can see that is the single circulation. Lizard and pigeon. So lizard, which is a reptile, pigeon is a bird. So bird which shows complete double circulation. Frog and crocodile. So crocodile exception, even though it is a reptile, that shows double circulation, complete double circulation. So answer for that is toad and lizard, which shows, what is it, incomplete double circulation. So fishes which shows single circulation. So closed circulatory system is found in. So we know that generally the closed circulatory system which will be there in the vertebrates, right? Generally that is the mainly includes vertebrates, and also that is there we mentioned that is in the annelids, right? Here if they are given mollusks, but instead of that they should give not all mollusks, only non-mollusks. That is cephalopod, not non-mollusk, that is cephalopods. So cephalopods are the closed, whereas the non-cephalopods are open type. So hence we can't select this. And amphibians and mollusks, here also we can say that it is not completely the closed. And in the arthropod and chordates, in arthropod, that is in insects, it is a open type. Insects, it is open type of circulation. So answer for that is annelids and chordates. Annelids and chordates are the correct answer. Closed circulatory system. Which of the following lacks a circulatory system? Whereas in case of lower organisms, if you consider that the planaria, which are comes under the flatworms and paramecium, generally we know that which will comes under the ciliates, that is a protozoans, ciliates. These are also the invertebrates. And jellyfish, if you consider that the octopus, right? So whenever we are going to say that the in this case example like jellyfish or octopus, right? So in which generally we'll say, that is in the non-cephalopod, right? So jellyfish, we can write here, these are non-cephalopods, not octopus, that is a cephalopod, right? Non-cephalopod, all of these which comes under there won't be any the circulatory system, they lack. That is, even though these are exchange through their body surface, diffusion through their body surface on. And we can also say that here we can say the flatworms and these things, the water which will be circulate, that is through their body surface. No circulatory system here, no circulatory system through their body surface. Directly it will going to get exchange the oxygen and carbon dioxide, and also system. In fishes, blood circulation is represented as, see here from the heart, I mentioned that heart to gills it will be pumped, and directly the pure blood from the gills to the body parts and impure blood to the heart. So if that is the case, only one time before reaching the the body parts, right, it will going to reach only once the heart, right? after impure blood which will going to come back. So that is the single circulation. Such heart is called as venous heart. Such heart is called as venous heart. So above blood, what is it? The flow of blood indicates what? Here we can say that this single circulation. It is not, what is it, the double circulation, or it is not incomplete single circulation, or it is, what is it, the incomplete double circulation. So here you can say that this single circulation. So that is what you have to keep in mind. That is means here it is not like that incomplete or complete. It is always, you can say, in this you can say complete result single circulation. Next, so in this case, blood and blood components, these questions they are going to ask. So what exactly the role of RBC? So RBCs helps in the transport of gases. RBCs helps in the, what is that, transport of respiratory gases. And WBCs provide immunity. That is, they play a major role in provide immunity, and what is it, in the mainly, what is it, the through the process of, some will going to show phagocytosis. So phagocytosis, and some which will going to directly they engulf, or you can say that they're going to show that the inflammation, right? So inflammation, and some will going to create, what is it, that is allergic. After allergic reaction there will be inflammation. So all these functions will be done by, so this is 120 days, and lifespan this will be here in this case some within a week, or you can say that the week to several month, right? So week, right, or you can say that this several month, right? So in this case, what is that, the platelets which are there generally, and the RBCs, right, in both the cases, platelets and RBCs, we won't see any nucleus, no nucleus. So these are the important thing, no nucleus in, what is it, the platelets, what is it, platelets and RBCs. So platelets are actually formed by, what is it, the megakaryocytes, the cells, megakaryocytes, and the fragmented part of this megakaryocytes. That is why we can say cell fragments. Platelets are cell fragments of megakaryotes. We are going to write like this, right? So generally with no nucleus, so these are the cell fragments of megakaryocytes. And next, let us come to this, the most abundant and least abundant. We discussed that most abundant is, what is that, RBC, that is 5 to 5.5, what is that, the million per cubic millimeter of blood, So RBC is the most abundant, and WBC, that is least abundant, that is 6,000 to 8,000, very less. Whereas platelets, we can say 1,50,000 to 3,50,000, which will be there per cubic millimeter of blood. So most abundant RBC, and then least abundant WBC, and then the medium number, we can say that the moderate number, that is platelets, right? So that is the answer for that is most abundant RBC, WBC. And here you have to keep in mind that monocytes are the largest leukocytes. Generally that will be, what is it, the 15 to 18 micrometer will be there. It is more than RBC, we can say more than RBC. So largest in the body is monocytes. And same like that, here we can also say that the smallest, if you come to this, what is it, in the smallest cell, that is the platelets, right? So generally in which case, in this case 2 to 3 micrometer will be there. So 2 to 3 micrometer, that is a platelets. RBCs, right, will be 7 to 8 micrometer. RBCs will be 7 to 8 micrometer. So that is why we will say medium size and bandaga, that is largest is WBC nalli, monocytes in particularly. And then what is that, after that you have to keep in mind that the WBCs, right, in the WBCs only, right, we can see that is the largest and here in the RBC, that is a medium size, and the less you can say, or you can say that the small size, that is a platelets, 2 to 3 micrometer, and RBCs are 7 to 8 micrometer, and WBCs are from the 8 micrometer up to the 15 micrometer. That varies. So next, a reduction in number of platelets. So we know that the thrombocytes, platelets are also called as thrombocytes. If it is reduced, that clot won't take place, and that leads to excess loss of blood. Clotting disorder, which also associated with, if there is vitamin K deficiency, we can consider that clotting disorder. Next, blood coagulation inside the vessel. Now, excessive loss of blood from the body. So here we can select excessive loss of if platelets count is less. Allergic responses are regulated by. So as we know that mainly allergic responses, right? We discussed that in case of the eosinophils. So keep in mind that these eosinophils, which are also called as acidophils, based on stain, we're going to classify. These are the acidic in nature, and neutrophils, both they are neutral. We can say that stain by we can say that this is a neutral, and basophils, which is a basic, right? So basic in nature. So hence the name we have, that basophils, eosinophils, acidophils, right? Stains for the acidic, and basophils for the basic, and neutrophils for both, that is we can say that the neutral, right? And here allergic reaction, that is eosinophils, right? And neutrophils, keep it in mind, that is a phagocytosis. Neutrophils, that is phagocytosis. And monocytes, that is we discussed that these are the the acts as a scavengers in the body, engulf, that is these are also called as, we can say, phagocytosis, but we'll say that scavengers. Why? Because this not only engulf the microbes, right? These only engulf the microbes, but the monocytes which are there, they act as a scavenger because they engulf the microbes and also cellular debris. This is the one difference we have to keep it in mind in case of monocytes. And this lymphocytes, which will be helps in the antibody production. So answer for that, allergic, that is eosinophils. Neutrophils, phagocytic, and then basophils, we know that heparin, serotonin, and the histamine will be produced. So next, let us move on to blood group system. As we all know that there are two blood group system, ABO and RH blood group system. We are familiar with this. So if RH antigen is present, that is RH positive. If RH antigen is absent, that is RH negative. Based on that, we will say A positive, the B positive, right? AB positive, like that we'll say, right? If RH antigen is present, we'll give for all those blood, we'll give positive. If no antigen, that is, we can say here, you have to keep in mind that first time it is seen in the rhesus monkey. It's a species, old age monkey. Till date we have, that is normally they are present surrounding to us. These are the rhesus monkey. First absorb this RH antigen absorbed in rhesus monkey. Even in this ABO system, we have the antigen. So just to understand that A blood group will be with A antigen, right? A antigen and B antibody, and same like that B blood group with B antigen and then A antibody. And here AB, if you consider both the antigen are present, A and B antigens are present here, and no antibody. This is, you have to keep it in mind that no antibodies are present in that A B blood group, and O, that is no antigen, nil, right? Here we can say that both A and B antibodies are present. So this is what, if no antigen, then we can donate to anybody. No antigen, hence O is called as universal donor. In that ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, O ಪಾಸಿಟಿವ್ ಅಲ್ಲಿ ಏನಿರುತ್ತೆ, RH ಆಂಟಿಜನ್ ಇರುತ್ತೆ, ಇದನ್ನು ಕೂಡ ಚೆಕ್ ಮಾಡಿಕೊಡ್ಬೇಕು. ಬಟ್ O ನೆಗೆಟಿವ್, ಇಟ್ ಇಸ್ ನಾಟ್ ಲೈಕ್ ದಟ್. ದಿ exactly universal donor is O negative. Same like that AB, both antigens are there and no antibodies. So So these antigens somehow they can, what is it, the compatibility can be seen, right? ಒಂದು ರೂಲ್ ಇದೆ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. ಈಗ ಪರ್ಸನ್ ರಿಸೀವಿಂಗ್ ಎ ಪರ್ಸನ್ ಬ್ಲಡ್ ಗ್ರೂಪ್ ಈಸ್ ಎ, ಇಫ್ ಇಟ್ ಹ್ಯಾಸ್ ಟು ರಿಸೀವ್ ಇವನಲ್ಲಿರೋ ಆ್ಯಂಟಿಜನ್ ಇರಲೇಬೇಕು ರಿಸೀವ್ ಮಾಡೋ ಪರ್ಸನಲ್ಲಿ. ಇಫ್ what is that, see, A can receive because there is a A antigen. In A we have A antigen, from A ಇಂದ ರಿಸೀವ್ ಮಾಡ್ಬೋದು. ಹಾಗೆ ಓನಲ್ಲಿ ನೋಡಿ ನೋ ಆ್ಯಂಟಿಜನ್, ಸೊ ದಟ್ ಈಸ್ ವೈ ವಿ ಕ್ಯಾನ್ ನೋ ಆ್ಯಂಟಿಜನ್ ಅಥವಾ ಸೇಮ್ ಟೈಪ್ ಆಫ್ ಆ್ಯಂಟಿಜನ್ ಇದ್ದರೆ ಮಾತ್ರ ವಿ ಕ್ಯಾನ್ ರಿಸೀವ್, what is that, we can receive, or else it is difficult, right? It causes, what is that, agglutination. So that is why there should be a compatibility. So AB is universal acceptor. So these are the things you have to keep it in mind. Next question, in the blood group, that is ABO blood group, that is based on the antigen, presence or absence of antigen. Discuss that. So AB can, what is it, they receive from anybody, and O can donate to. So this here, one thing you have to keep in mind, A and then B and O, that is by discovered by Karl Landsteiner. Actually we'll say that is discovered by Karl Landsteiner, ABO system. But keep it in mind here, ABO and that is AB separately discovered by De Castello and Stini. So these are the two, what is it, the person who gave that blood group system, right? So that is a rule to keep it in mind. Let us move on to the next question. A person with the blood group A requires a blood. So the blood group which can be given, ಯಾರಿಂದ ತೊಗೋಬೋದು, ನಾನು ಹೇಳಿದೆ ಎ ರಿಸೀವ್ ಮಾಡ್ತಿದೆ ಅಂದರೆ ಇಲ್ಲಿ ಎ ಆ್ಯಂಟಿಜನ್ ಇದೆ, ಎ ಆ್ಯಂಟಿಜನ್ ಯಾರ ಹತ್ರ ಇದೆ ಅವ್ರ ಹತ್ರ ತೊಗೋಬೋದು, ದಟ್ ಈಸ್ ಎ ಇಂದ ತೊಗೋಬೋದು. ಸರ್ ಎ ಬಿನಲ್ಲಿ ಇದೆಯಲ್ಲ, ಬಟ್ ದೇರ್ ಇಸ್ ಅ ಬಿ ಆ್ಯಂಟಿಜನ್, ಸೊ ಎನ್ಸ್ ಇಟ್ ಮೇ ಕಾಸ್, ವಾಟ್ ಇಸ್ ದಟ್, ದಿ ಕೋಆಗುಲೇಷನ್, ದಟ್ ಈಸ್ ಅಗ್ಲೂಟಿನೇಷನ್, ಬ್ಲಡ್ ಕ್ಲಾಟ್ ಆಗುತ್ತೆ ಒಳಗಡೆ, ಆ್ಯಂಟಿಜನ್ ಆ್ಯಂಟಿಬಾಡಿ ರಿಯಾಕ್ಷನ್ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ಈಸ್ ವೈ ವಿ ಶುಡ್ ನಾಟ್ ರಿಸೀವ್ ದಟ್, ರೈಟ್? ಎ ಇಂದ ರಿಸೀವ್ ಮಾಡ್ಬೋದು, ಆರ್ ಎಲ್ಸ್ ವಿ ಕ್ಯಾನ್ ಸೇ ಫ್ರಮ್ O is a universal donor because no antigen at all, no antigen. ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, ಬ್ಲಡ್ ಗ್ರೂಪ್ಸ್ ಡೊನೇಟ್ ಮಾಡಬೇಕಾದ್ರೆ ಇಫ್ ಇಟ್ ಈಸ್ ಅ ರಿಸಿಪಿಯೆಂಟ್ ಆ್ಯಂಡ್ ಫ್ರಮ್ ದಿ ಡೋನಾರ್, ದೇರ್ ಮೇ ಬಿ ಆ್ಯಂಟಿಬಾಡಿ ಫ್ಲಕ್ಚುಯೇಷನ್ ಇದ್ದರೆ ನಡೆಯುತ್ತೆ, ಬಟ್ ಆ್ಯಂಟಿಜನ್ ಫ್ಲಕ್ಚುಯೇಷನ್ ಇದ್ದರೆ, if you're going to give that antigen to the body, to the recipient body, recipient antibodies will going to act, what is it, the vigorously, immunity they're going to increase and they're going to create a problem, right? Hence there should not be difference in the antigen, So what is that? He can be received, what is that, from A and O. And which of the following blood group, universal donor and universal acceptor? So universal donor, we can donate to anybody, no antigen, that is O, right? So in this case donor, and universal, what is it, the recipient, AB. So we know that is based on. So here this is also like, what is that, the if you are donating blood to A positive, that you should take that from the A positive, that will be, there will be antigen, which is that antigen, RH antigen, that is RH positive, right? If it is A negative, right, you can't receive from the A positive. Keep in mind here RH antigen is there and here no RH antigen, that is also will going to, what is it, they create, or what is that, incompatibility. So if RH negative person, see, assume that if RH negative person, so negative person means no antigen, right? No antigen, RH no antigen. If you want to receive, the RH negative person donates blood to RH positive, right? Negative means no antigen, here RH negative, that is why we can say O negative can donate. Even in this also no RH antigen, you can donate to anybody, you can donate to A, you can donate to B, you can donate to AB, and you can also donate to O, right? But not largely to the O positive, but here O negative easily can receive, even for that O positive also you can donate. So here, what is that, RH negative person donate blood to RH positive person for second time, then what happens? So here just we are not discussing about erythroblastosis fetalis, it is not like that, what is it, where the person will going to they receive the blood, like mother and the fetus, as we know that is in case of erythroblastosis fetalis. Right? Here RH negative person, keep it in mind, negative person andre no antigen, right? No antigen, and donated blood to antigen. So here that if no antigen, no reaction will be takes place. So that is why nothing happens to the RH positive person. So RH positive person will die. How? Here no antigen, how reaction won't takes place, you won't the die, right? Generally the RH positive person, right, won't die. And next, RH negative person starts reacting to RH positive, start reacting. As RH negative person is donating, how come RH negative person will going to start react, right? Donate ಮಾಡಿರೋರು ಹೇಗೆ ರಿಯಾಕ್ಟ್ ಮಾಡ್ತಾರೆ ಇಲ್ಲ. ಸೊ and then nothing happens to, and you can say here, we can also say that RH negative person blood, if blood start reacting in the RH positive, here keep it in mind, to start react there should be antigen mismatch. ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, antigen mismatch ಇದ್ರೆ ಮಾತ್ರ. If I am donating blood, I won't have antigen, I am donating blood to the person, whether it may be positive or the negative, right? Here no antigen in my blood and no reaction, nothing will going to happen to a RH positive person. Next, erythroblastosis fetalis. So it is, which is also called as hemolytic disease in newborn. Hemolytic disease in newborn. What is that? Hemolytic. I will tell you why. Because here the lysis of blood cells takes place. Hemolytic disease in newborn. In newborn. So that is what we can say here in this case. That is, to prevent that, we're going to use ROGAM. ROGAM is a injection. What is, just we'll take this. If RH, what is it, the negative mother, RH negative mother will going to marry RH positive, what is it, the man, that is the woman. If she marries RH positive man, then we can say here will be, what is that, antigen. So when the pregnancy, when she's going to maintain a pregnancy, half of the chromosomes will be received, so that they're expected that the fetus may be with RH positive. So mother blood is RH negative, right? In the first pregnancy, the placenta will going to show little, what is it, the impermeable, and where the rupturing takes place, very small quantities of antigen will be there here, right? That very small quantity will going to expose with the mother blood. So in the mother blood there will be antibody, keep it in mind, mother blood ಅಲ್ಲಿ ಆ್ಯಂಟಿಬಾಡಿ ಇದೆ. ಇದು ಈ ಆ್ಯಂಟಿಜನ್ಗಳನ್ನ ರೆಕಗ್ನೈಸ್ ಮಾಡಿ ಇಟ್ ವಿಲ್ ಗೋಯಿಂಗ್ ಟು ಶೋ ಸ್ಲೈಟ್ ರಿಯಾಕ್ಷನ್, ಬಟ್ ಇಟ್ ಕಾಂಟ್ ಬಿ ಏಬಲ್ ಟು ಶೋ ವಿಗರಸ್ ರಿಯಾಕ್ಷನ್. So RH sensitization will be less there. Mother's blood during the first pregnancy, it is very less, right? I will tell you that is because, keep it in mind, simple thing, IgM initially will going to produce, because it is a less potential, can't pass through, less potential can't pass through placenta. ಅಷ್ಟೇ. ಸೆಕೆಂಡ್ ಟೈಮ್ ಏನಾಗುತ್ತೆ, in the second pregnancy where the what is it, the fetus with RH positive with antigen, ಇಲ್ಲಿ IgM which will be converted into IgA, and this is a placenta. IgA can easily pass through placenta, that is to the mother blood. So now it's very important thing, these antibodies can pass through placenta. When they can pass, very very important, mother's antibody will going to act, what is it, the vigorously, and that will going to, what is it, the catalyzes the blood, and it will going to lysis. That is why it is called as fetalysis. First it lysis the, what is it, the this will going to, as they will going to pass, but lysis and embryo in which the RBC will be there, that complete antigen will be there, these antibodies will going to kill, so destroy RBC completely. So that is what the thing which will going to takes place. So in the mother's blood, so these things will going to takes place. Hence the person will going to marry, what is RH positive, what is it, the man. So during the pregnancy should undergo, what is it, the test, right? In the second pregnancy, RH antibodies will be going to increase, right? Can leak, RH antibodies can leak into the, what is it, the where the fetus development takes place. And there will be in the fetus, we know that there will be antigen, right? Because Rh positive it is. So these antibodies once enter, they're going to destroy the fetus. Hence it is called as destruction of red blood cells, what is that? Hemolytic disease. Hemolytic. So mainly RBCs will be, what is it? Destructed, or erythroblastosis fetalis. Now, disease erythroblastosis fetalis in, what is it, the of human baby is due to, right, incompatibility of blood groups of embryo and mother, right, incompatibility of blood group of embryo and mother, right. Generally in this case, no, what is it, the if there is an incompatibility in the blood group of embryo and mother, it won't show. But when it arises suddenly in the first pregnancy, we can't see that, right. In the second pregnancy, consequence pregnancy by the same, that is if they are the couple, right, so then we'll say that is incompatibility of blood groups of, what is it, the couples. That is what we can say expected thing, Rh positive 50%. So then the couple will going to have the progeny, right. The progeny may be which will be going to with Rh positive, and the mother's blood, right, will be Rh negative. So this will going to create a problem. Here this side in the mother there will be antibodies, and here there will be antigen. So antibody antigen reaction destructs the RBC, hence it is called as hemolytic disease. Important function of the lymph. So whenever we're going to discuss about this lymph is a tissue fluid with lymph vessels. See, keep it in mind, there will be, what is it, the body tissue cells, right, there will be body tissue cells, right. In between these vessels, the capillaries which will going to run, right, there because of the smallest, what is it, the diameter, the nutrients will going to leak out. If it is accumulate for longer duration, it may cause problem. Hence it will be collected by, what is it, the smaller lymphatic vessels, and these vessels, when they're going to, what is it, they collect this, the interstitial fluid which is present between the tissue, and that will be drained to the smaller, what is it, the smaller lymphatic vessels. These are the lymphatic capillaries into lymphatic vessels, and lymphatic vessels finally open into the larger vein. So that is what the role of, what is it, these acts as a, lymph acts as a middleman of the body. Middleman, whatever the things will going to leak out here, that will be collected and that will going to pump. So this is what you have to keep it in mind. What exactly there will be, what is it, we can say that in the lymph no RBC and no platelets. There will be WBC in the lymph, no, what is it, the RBCs, red blood cells, and what is it, platelets. They can't pass through that capillaries, but there will be WBCs, white blood cells. So this is what, so lymph, more carbon dioxide will be there, because in between the interstitial tissue fluid is there, no, there will be a high carbon dioxide, and then WBC will be there, no RBC and platelets. So this is one, WBC will be there, but little, not compared to the blood, not as compared to the blood, it won't be more, right. So lymph, which mainly helps in, what is it, mineral distribution, right. Generally it will take that the leak minerals, or you can say that the, and sometimes it collects a waste also, we can say it collects waste and leak minerals, and that will be sent back to the body circulation. So next, megakaryocytes, right, we can say that the K also, better to say K, megakaryocytes produced. We know that megakaryocytes are the cells which will undergo, what is it, generally fragmentation. The fragmented part of those megakaryotes are called as thrombocytes, that is blood platelets, right. So keep it in mind that these leukocytes are produced in the bone marrow, right. So this is very, very important, right. Leukocytes, we can say red bone marrow, and I can say that the lymphocytes, these are also generally lymphocytes which we can consider that thymus and lymph nodes, produced in thymus and lymph nodes production I am telling, right? So lymph nodes ಅಲ್ಲಿ produce ಆಗುತ್ತೆ ಅಥವಾ thymus ಅಲ್ಲಿ ಆಗುತ್ತೆ. ಹಾಗೆ monocytes, we can also say that these monocytes which will be produced in the lymphocytes and also in the spleen, lymphocytes and spleen. So these which are there, thymus and spleen, which are the lymphoid organs mainly that helps to produce the lymphocytes and monocytes. Whereas RBC, we know that RBC in the red bone marrow, that is RBC, and then WBC, all other things RBC in WBC except these monocytes and lymphocytes, monocytes and what is that, this is lymph nodes, lymph nodes. Monocytes are also produced in lymph nodes, lymph nodes. Both monocytes and lymphocytes produced in the lymph nodes, whereas lymphocytes produced in the thymus and then monocytes produced in the spleen, RBCs, and then all other granulocytes, right? All other granulocytes produced in red bone marrow, red bone marrow ದಲ್ಲಿ produce ಆಗ್ತವೆ. So these are the things you have to keep in mind. 1 cubic millimeter of blood has how many, what is it, the blood platelets? That is 1 lakh to 3 lakh 50,000, 1 lakh 50,000 to 3 lakh 50,000. What is that, the 3 lakh per cubic millimeter of blood, we can say. So here this million means 10 lakh, so that is wrong. 1,500, 3,000 wrong. 10 to 15 lakhs wrong. That is 1 lakh 50,000 to what is it, 3 lakh 50,000 cubic millimeter of blood, the blood platelets count in the normal healthy person. A doctor suggested to a couple not to have more than one child. ಯಾರಿಗೆ ಹೇಳ್ತಾರೆ, ಸಜೆಸ್ಟ್ ಮಾಡ್ತಾರೆ? ನಾವು ಹೇಳಿದೆವು ಫಸ್ಟ್ ಪ್ರೆಗ್ನೆನ್ಸಿನಲ್ಲಿ ದಟ್ ಇನ್ಕಂಪ್ಯಾಟಬಿಲಿಟಿ ಇದ್ದರೂ ಕೂಡ ಲೆಸ್ ವಾಟ್ ಇಸ್ ದಟ್ ದಿ ಎಫೆಕ್ಟ್ ಇರುತ್ತೆ, ಲೆಸ್ ಎಫೆಕ್ಟ್. ದಟ್ ಈಸ್ ಇನ್ ಕೇಸ್ ಆಫ್ ಎರಿಥ್ರೋಬ್ಲಾಸ್ಟೋಸಿಸ್ ಫಿಟಾಲಿಸ್. So that is the one you have to keep it in mind. When Rh negative, what is it, the female marries Rh positive male. So here we can say that is, what is that, Rh positive male marries, so Rh negative female, that the second time to have a pregnant, what is it, the child, that is difficult. We can, after as soon as give birth to first one, right, they're going to give the injection, that is anti-D injection. Erythroblastosis fetalis, to prevent that they're going to give ROGAM, or you can say anti-D injection. So ROGAM, just you keep it in mind that within 72 hours, within 72 hours of the first child, because even though after late the exposure which will be takes place, for that they're going to show reaction that prevents that reaction, so that will be completely neutralized. So second time, what is it, the pregnancy can be maintained. ಇಂಜೆಕ್ಷನ್ ಕೊಟ್ಟ ತಕ್ಷಣ ಈ ರಿಯಾಕ್ಷನ್ ಏನು ಸ್ಟಾರ್ಟ್ ಆಗಿರುತ್ತೆ ಫಸ್ಟ್ ಚೈಲ್ಡ್ ಆದಮೇಲೆ ಅದನ್ನು ಸ್ಟಾಪ್ ಮಾಡಿಬಿಡುತ್ತೆ. ದಟ್ ಈಸ್ ವಾಟ್ ROGAM, ದಟ್ ಈಸ್ RH gamma globulin, RH gamma globulin. Or we can also say that generally, so in this case, to prevent that the death of the second this one, they're going to give the injection, right? So within 72 hours after, sorry, mother after, what is it, or within 72 hours after first childbirth, within 72 hours they're going to give injection. Why this is called as anti-D? RH that is also called as anti-D. RH antigen is also called as RH antigen, which is also called as anti-D, right? Antigen D. Hence we will say that is, what is that, the anti-D injection against antigen D. RH antigen, which is also called as D antigen. So against that, the injection which will be given, that is anti-D injection. Next, which of the following are cell fragments? That is we discussed. That is thrombocytes or blood platelets are the fragments of megakaryocytes. RH factor is concerned with blood grouping. It derives its name from. So generally, I mentioned that not in the man, rat, or you can say the chimpanzee, monkey, that is rhesus monkey. Don't get confused. Rhesus andre adondu species actually, right? Commonly present seen anta, commonly present seen adu, right? Next. So in the next class we can take up the heart structure. So this we can stop. In the next class, in the second part, we can take the structure of heart and then the blood clotting related things. Thank you.